then our first speaker for the first lecture is Jan Rudolph. And he's a now PhD student in Max Planck Institute of Biochemistry under the supervision of Professor Dr. Jürgen Cox. He studied his bachelor's degrees in Tübingen, and then he moved to Israel, and he studied in Tel Aviv University, where he actually worked on a phosphorus. And in 2016, he joined to our lab, and since then he is working on little more terms, and he's involved in the Perseus development. And today he's going to tell us about the dependent peptide search for unbiased search for PTM. Sorry.
Thank you, Shule, for this kind introduction, and good morning to everyone. I hope you're all energetic and ready to learn something about PTM analysis, and more specifically, how to use your super accurate mass spectrometer to identify PTMs that no one's ever seen before. So, in this talk, we will cover the dependent peptide search, and I will try to focus on 3 points. So first, that you understand the differences between the dependent peptide search and the standard search, which is how you usually run MaxQuant, and also understand the advantages and applications. And lastly, I will also quickly show you how you can use it for your own analysis. Okay, so the main motivation is, can I use my shotgun proteomics data to identify new and unknown protein modifications? Now, the mass spectrometer is an excellent tool to study post-translational modifications, because every modification on amino acids introduces a mass shift on the peptide, which can be detected by the mass spectrometer. With modern instruments, we have really high sensitivity, so we can even uncover sub-stoichiometric modifications and analyze them. And due to the high mass accuracy, even modifications which have the same nominal mass can be distinguished. So due to the fragmentation on the MS2 level, we can also localize the modifications inside the peptide. So there are different kinds of modifications that you can study. They could be co-translational or post-translational, but they could also be induced during sample preparation, as Tammy told you in the sample prep talk. And we can also try to find sequence alterations, not with the same precision as the proteogenomics approach that Prabha presented, but we might still find some interesting amino acid substitutions. Okay, so let's see how we generally search for modifications with MaxQuant. So inside your MaxQuant group-specific parameters, there's this modifications tab, and it has this table where all the variable modifications are listed. And these are exactly all the modifications that you configured yesterday in Andromeda. So let's say you're interested to study phosphorylation. So you would then select your phosphor-STY and move it to the right side of the panel, and now MaxQuant will try to find all your phosphorylations in the data set. How will it do it? It will add them to your sequence database, so all phosphorylated peptides will be in there. Okay, so this is like a very brief overview of how the peptide identification works in MaxQuant. In the next talk, Petra will give you more details on this. But let's go along the pipeline. So we start with all our observed peptides, which are our MS1 features. So we have all the masses. And then we will try to match our observed mass to the theoretical mass, which is written in our protein database. So if we have an identical match within a small margin of error, we will consider this peptide for identification on the MS2 level. So let's see what happens when we have a modification that we did not specify in the variable modifications tab. So this unknown modification will introduce a mass shift on the MS1 level, and therefore we will not be able to match it to anything that we see in our database. And subsequently, this peptide will be excluded from the search. So we will not be able to identify it. So dependent peptides, they are synonymous for peptides with an unknown modification and therefore with an unknown mass shift. Now, these peptides are not identified during the conventional database search, but we can try to still identify them using the dependent peptide approach. So instead of trying to match them into the database, we will try to match our unidentified peptides into our already identified peptides under the assumption that maybe our peptides with unknown modification, our dependent peptides, they are just a modified version of a peptide that we were already able to identify. So this is where the name dependent comes from, because we will only identify dependent peptides if we identify the unmodified counterpart. And this concept was introduced by the Zubarev lab already quite some years ago. Okay, so dependent peptides, they can originate from different scenarios. So dependent peptides might just bear unknown modifications, but also protease cleavage can lead to shortened peptides, which you can think of as just a big minus delta mass. And also amino acid substitutions will just be seen in the mass spec as a change in the mass of the peptide. So let's see how this works in MaxQuant. First, let me briefly review the target-decoy search for the normal peptides. So here you have 3 populations of peptides. On the right, you see all the observed peptides. So these are the ones that you measure in your instrument. And on the left side, you see theoretical peptides on the top, the good ones that you have in your database. And on the bottom, the decoy peptides, which we will use for our FDR estimation. So now we said that we will match observed peptides to theoretical peptides if their mass is identical. So in our entire protein database, in the theoretical peptides database, there will be only few matches, and hopefully even less on the decoy side. So now how do we decide which of these matches we take? We calculate the Andromeda score for every pair, and then take the best one. Okay, so how does the scenario look for dependent peptides, which we said do not rely on the database, but rather on the already identified peptides? So it's the same picture, but on the right side now you have the unidentified peptides. On the top left, where you used to have the database, you now generate a new database from the already identified peptides. So we replace the theoretical peptides with the already identified. On the top, on the bottom, you have a new set of decoy peptides derived from these identified peptides. Okay, so now that we cannot restrict ourselves, to just mapping between peptides with identical mass, but rather if we consider an unidentified peptide and match it to one identified peptide, we will always have this mass shift, which we can calculate. And in order to find the best match in our identified peptides, we actually have to do this for all peptides in our database. So you can already see that there's a lot of stuff going on, and from all these we have to find the best match. So this is quite computationally intensive, that's also a reason why the dependent peptide search takes quite some time. And I hope from this huge amount of errors you can appreciate why we cannot just use the entire database of theoretical peptides that we used in the regular approach. We have to restrict ourselves to some set of peptides where we are more confident that they are actually in our samples. So this is why the dependent peptide search is put in place to put this assumption on the peptides that they are not just showing up in our sample, but are rather a modified version of a peptide that's already there. Okay, so let's look a bit deeper. Petra will again cover Andromeda, but I just want to mention that to score our matches, we actually use a modified version of the Andromeda score, which is just giving a probability to finding k out of n theoretical peaks. And here, since we are not matching to theoretical spectra, but rather to our previously identified spectra, we have to switch out the theoretical peaks with our observed peaks. And similar to the PTM localization, we will also do the same in the dependent peptides, where we try to apply the mass shift to all different positions. So here, this is one example peptide, where we just put the delta mass on all the different positions and then calculate the Andromeda score. And from all these scores that we have for the different position, we can also calculate the localization. So I will make this clear in 1 second. So our decoy peptides also have to be generated now from our set of identified peptides, and we use a similar approach where we revert the peptide that was identified and swap the lysine with an arginine to keep the same mass. And then we will have the same number, length distribution, amino acids, and so on in our decoy database to keep it as similar as possible to our identified peptides. And we can use this, of course, then to calculate our FDR separately for our dependent peptides. So let's look at the quick example. Here on the right side, you can see a spectrum of a peptide which we were not able to identify. But we looked in all our identified peptides, and we found the one that matched best with the Andromeda score. And this is the one identified peptide that you see on the top. And what we also know from the MS1 level is that these peptides are somehow different by a certain mass. So now what we can try to do is to see where in the peptide would we best put this mass difference so that the spectra are most similar. So here I will show an example of putting, for example, this delta mass on the first amino acid, and you can see that we can generate this Y-ion series, which we can use for the matching, and we will put a mass shift on all the Y-ions, which you can see in orange in the first position. Now for this series, we can calculate the Andromeda score on the MS2 spectrum, and then go on to the next position inside the peptide. So here we put the mass difference on the second amino acid, and again we generate this Y-ion series. And you can see that now the very first Y-ion does not bear any mass difference, but all others do. And again we can calculate the score, and so on we can go through the entire peptide. So here I show only the Y-ions, but the B-ions are also considered in MaxQuant. So now we would choose the best score of all these possible positions as our localization of the modification. So here you can see an example. This is the base peptide, which we were able to identify during our regular search. And on the bottom you can see the dependent peptides, which we were able to uncover using the dependent peptide search. Now if you compare the Y-ion series, you can see that the Y1 ion is actually identical between the top and the bottom spectrum, but starting from the second Y-ion, there's a mass shift along the entire series. And due to the size of the mass shift, we can say that there was a lysine acetylation on this specific amino acid. And since the spectrum is quite nice in this case, we can be very, very sure about the localization of the acetylation. So we cannot really explain it any other way but that it's actually located on this very first amino acid. Okay, so let's again review the differences between the standard search and the dependent peptide search. in the standard search, we try to match our observed fragmentation spectra against the theoretical spectra from the protein database. While in dependent peptide search, we try to take the unidentified fragmentation spectra and match them to the already identified spectra. If we look at the modifications that we can study, in the standard search, only N-terminal acetylation and methionine oxidation are considered. And this is mostly due to the speed of the analysis, and especially since just including more and more variable modification will really blow up your FDR because it goes with the database size. In the dependent peptide search, we can actually search for an unlimited amount of modifications, and we are also not restricted by any predefined list of modifications. Rather, every mass shift will be considered. So I already said that in the standard search, we might have FDR problems if we take too many variable modifications. But on the dependent peptide level, we have a separate FDR control for the dependent peptides. The standard search has a huge advantage that it's quite fast, while the dependent peptide search really can take a long time. Okay, so now that we covered the theory, let's look at some examples and where you can apply dependent peptide search in your projects. So this first example is a SciLec data set from mouse embryonic stem cells. And in the standard search, only the standard variable modifications were selected. And the identification rate on the MS2 level was about 52%. So now we can run the dependent peptide search and see if we can still identify more of these MS2 spectra. So here in the pie charts, you see the different identifications. And you can see that exactly this 52% identified by the standard search, which are mostly unmodified peptides. So this is as you expect. Unmodified peptides and exactly the modifications that you specified in the variable modifications. So 13 additional percent can be identified by dependent peptide search, and so we will take a closer look at what kinds of modification we can find there. So if we first look at the mass difference that we find between dependent peptides and their unmodified counterpart, we can see that it ranges over a big range of masses, and this is on log scale, so most of the modifications are actually in this minus 100 plus 100 range. So if you zoom in, you can really see the distinct peaks of the different modifications. And I just want to highlight here on the left side of the bottom panel that sulfur and O2, which have the same nominal mass, are two very distinct peaks due to the high mass accuracy. So if we look at if we try to annotate all these modifications, we can see many different modifications present in our sample. And I just want to pick out one group for this example, which is this new peptide group. And these are exactly peptides which mostly lie in this minus 500 or minus 1,000. So these peptides, they lost a lot of their mass. And you can think of this N-terminal shortened and C-terminal shortened peptides as ones that were degraded by additional protease cleavage. So since we have the sequence of our peptides that lost so much mass, we can also look and try to understand which protease actually was responsible for this. So in this sample, there was additional chymotrypsin cleavage found. Okay, on to a second example. Here, Eva Kallhauer, which is in the department, studied E. coli, and she actually, with her collaborators, found a very new arginine modification, a glycosylation that was never reported before in E. coli. So you can see here this rhamnosylation. And what was super cool is that they actually found that this novel modification also links to the pathogenicity phenotype of this E. coli. Okay, a third example is looking at histone modification. So this is based off just the standard HeLa data set, so it's not even anything fancy, but what you can do is you can just filter, for example, all the proteins that you found, peptides that you found for histone peptides. And then you can look at the different modifications. And of course, you see the methylation and these things. And the next step would be to see, okay, maybe I identified something new. So you can go, for example, to UniProt and try to see if the annotated modifications are already there. And for example, so this one was already annotated in the data set. But in the very same data set, there was another dependent peptide bearing this acetylation on this lysine that was actually not reported before. So even in routine data sets with dependent peptide search, you might see something that no one's ever reported before. Okay, so I think that's really exciting. So let's see how we can use it. It's actually that simple. You just, like many things in MaxQuant, you go to the global parameters in the advanced identification tab, and you just hit that button. So I'm not saying everyone should always do it. It really takes a long time for processing. So you should only do it if you are really interested in those modifications that are non-standard. So you have to see how it fits your project. But I also have to say that checking this box will not in any way impede the results of the standard search. So this is a process which is independent of the rest of your analysis. Okay, so here I will show you how to load the dependent peptides table into Perseus. So Perseus is like built with plugins, right? So there's a plugin for reading the dependent peptides table because it's a bit complicated. And what you can see me doing here in this short clip is actually downloading 2 plugins that we need. So you go to the Perseus documentation page and you try to find the plugin store. And once you get there, there are 2 plugins which you need here, which is this plugin interop and the plugin dependent peptides, which you can then download. So once you have those zip archives, you just have to unpack them, which you can see me doing here. So, I mean, the purpose of these videos is basically to show you that it's super easy, but that there are like a few steps involved, and it really takes only a minute. Okay, so how do you install plugins? You download them, and then you drop them in the bin folder, and that's all you need to do. Perseus is smart enough to see which DLLs are in the same folder, and then will load them automatically. Okay, so now that you installed your plugins, you have to load the all peptides table and the experimental design table, and then you have to practice something that everyone has to practice that does dependent peptide search, is patience. So the all peptides table, which is the basis for the dependent peptide results, is actually one of the bigger tables in the MaxQuant output. So this is several gigabytes of data that Perseus has to digest now. But at some point it will show up, and you will see your table with all your experimental conditions and the dependent peptides. And yeah, Windows is bugging me here, but I hope you can see that this is definitely something that everyone can do. And yeah. So which kinds of modification can you find in your table then? So there are obvious ones. So there will be a dependent peptide modification column, and it will have methylation written there, so that's good. There might also be this comp sections, which is just the elemental composition. And there can also be unmodified peptides. So this is nothing to be afraid of. We know unmodified peptides, and why do they show up in the dependent peptide search where we were looking for modifications? It's because the dependent peptide search is very sensitive. And maybe we have the same peptide already in our sample, but we are not able to assign it all its MS2 fragments. So now there's a new MS2 event that was not scoring high enough in the standard search, but in the dependent peptide search, it will be assigned to the correct peptide. Then there can be also peptides which have this dot annotation. So this means that the peptide was extended in C or N terminal. And there can also be this no entry sections. So the no entry just means that, okay, so where do all these names come from? These names come from the Andromeda configuration. So even though you didn't choose these as variable modifications, when the output is generated, MaxQuant will try to assign all these delta masses to a human readable name, and we'll look up in the Andromeda configuration for this. So maybe you have a no entry here, but you will have the mass difference, and maybe you will know which modification this is, and you can actually then configure Andromeda to include this modification and even study your sample then using this maybe novel modification as a variable modification to really have the sensitivity to pick it up. Okay, and so I think with this, we all know what we have to do now, and I would be very happy to take questions.
Do we have any question? Oh.
I think we have.
Please go ahead.
Okay. Hi, good morning. I have actually two questions. First, these are not fixed modification. They're not taking consideration for quantification afterwards, right?
So the dependent peptides, they are quantified.
For the protein groups, they are used
no.
for quantification. Not
No.
used at all.
But you will have peptide level quantification on these dependent peptides.
On the peptide level.
Exactly, yeah.
Okay. And the second question is, I don't really understand because you said you have to look for the dependent peptide based on the peptides that you found already. But this newer peptide will have an unknown magnitude,
Yes.
which means all the spectrum will be shifted. So you showed us an example of a lysine modification, but out of 10 ions Y series, 9 of them were shifted. So how can this match to the peptide if all the spectrum is shifted?
Okay, so I tried to show this in the example where I had the base peptide and I put a modification on every amino acid. And so what's done internally is, this is more of a thought experiment. You put the amino acid on the residue and you look how theoretically now does my Y look. So I put it on the first amino acid, I see all my Y ions are shifted. I put it on the second, and then maybe the first one is not shifted, and all the others are shifted. And this exactly will then match perfectly to my experimental spectrum that I saw.
Yeah, that's for the positioning of the modification, right? But how do you identify that this is the size of the modification and this is exactly the same peptide as before?
Yeah, so I try to explain this here in a two-step process, but this is actually done in one step. So just to make it less complicated, I try to split it up. But exactly when you try to, so we have this picture with all the arrows where we try to match our identified spectrum, unidentified spectrum in, and there we actually consider all different positions inside the peptide as possible locations of the modification.
Okay, thank you.
Right, something related to that question. So if the peptides is enabled, are the additional peptides taken into account for the protein roll-up later on, or is it just that they are an additional in this peptide list that you can load?
So you can correct me if I'm wrong, but I think they are not taken into consideration for all the standard search results.
So I would have to take them into account in the second search, just knowing that they are there, so that I can take the variable modification maybe.
I think the practical approach is to not worry about the dependent peptides in they are like a separate thing. Like you analyze the data regularly with the results that you always get. And the dependent peptides are just additional information if you want to dig deeper in your data in this direction.
Hi, up here. So I understand that when you replace the theoretical peptides with the identified peptides, that of course that improves your or gives you a better FDR. But in case, for example, of a homozygous mutation, point mutation, or a modification that's so abundant that basically there is no normal peptide left, then that you would miss, right?
Yes, exactly. I mean, this is inherent to the dependent peptide idea that you need this unmodified counterpart to be in your sample.
And did you think of a way, or is there a way to maybe to look for unidentified peptides that are so abundant that it's likely that you assign a certain likelihood to them to not be some trash, basically, but actually have biological meaning? Would that be a way of modifying or complementing this approach, or is that basically also in terms of FDR not possible?
Yeah, so I think it's very difficult without knowing the sequence of the peptide to really say much about it. I think you're best, if you're an expert in a specific domain and you are expecting exactly specific mutations maybe from the proteogenomics, then you will have this either encoded in your database directly or with the variable modifications. I think these are the tools that are most robust for the analysis.
But in principle, you know the peptides, but it's like you just don't have your identified peptides. It's basically, if you look for a point mutation, it's not a totally different peptide. It's just also one minor modification on a peptide that's known in the database. But still, that's statistically probably not giving you enough.
Yeah. I
I
think because you really run into problems then, because if you just try to match it to everything, you will also have this huge set of decoy peptides, and you will most likely also have some explanation with the decoy peptides, and then it will not pass any FDR threshold.
Okay, thanks.
Yes.
My question is about mass shift determination. So how do you specify the delta M? Does it scan for all the possible delta M's between the unidentified features and the already identified peptides? Because it seems like a huge computational work. Yeah. And if it doesn't, how does it guess or figures out what delta M is more likely to appear?
I think this is easiest to answer if you think about the MS1 features. So the dependent peptide, you know the mass from the MS1 feature. And now you look into your identified spectra, which also have MS1 features. So now you just compare the masses of the two, and you immediately get the delta mass. Okay, so it's not scanning, it's just going through the list and calculating it pairwise.
So if I want to repeat the dependent search, dependent peptide search, is there any recommendation from which partial processing I can start, or I need to repeat the whole entire search?
Excuse me, I had a hard time.
Okay, so if I want to repeat the search with dependent peptide option, yes, is there a partial processing step that you recommend, or we need to repeat again?
I don't think there's any. I think you have to just redo it. I think
redo
it's
it.
the easiest.
Oh, thank you.
Thanks for the interesting talk. My question may sound naive, but when you're making these decoy peptides, is there any chance that this actually will be a peptide that's found in proteins, or because you're reversing it and you're adding the arginine? And
yeah. I guess by chance this could happen, but it's really unlikely. And I think it's just the approach is analogous to the way that we do the protein database, where we also reverse the sequences and then do the in silico digest. Right? So here we also have exactly the same assumption that this reverse peptides are also not actually occurring in the proper proteins.
Okay, thank you.
So what for do I have to load the experimental design into Perseus?
Excuse me?
What for do I have to load this experimental design file into Perseus?
Okay, so the the dependent peptide search, it operates on the raw file, so since it does not do any protein modification, there's no need for to see the experimental design of MaxQuant, so it's just so you have the columns with your different peptides listed there. So it's basically, so you can just load the all peptides table using the green arrow that you use for your protein groups table. And it's just a format that I think is a lot harder to parse and to understand.
Thank you for all. Maybe we can continue later.
Okay.
Sorry.
Yeah.
Okay.
So, in the current setting, all hypothetical modifications are permitted for the dependent peptide. Wouldn't it make sense to exclude some very unusual peptides to limit or to retrieve more peptides due to the FTIR control?
So your idea is to put more restriction on the database for the dependent peptides, so on the identified ones?
As in the conventional standard search.
Yeah, so I don't think that we tried anything in this direction. It could be helpful, but then you would have to be very precise about how you choose your peptides, and I think it's hard to find like general rules which are helpful.
And how many amino acid extinctions are permitted?
I'm not sure if there are limitations. I think whatever delta mass you have, Andromeda will try to match the modification to it. If it's multiple amino acids, then it will be multiple amino acids.
But that means that a short peptide with variable numbers of edited amino acids will create any amino acid combination from that.
Yeah, so, but then the best scoring one will win.
Question concerns the identified counterpart. The identified counterpart must be an unmodified peptide, or can it be also a modified, for instance, methionine oxidized peptide?
I think it can be any identified one, so it can be modified.
And the delta mass will then be related to the unmodified counterpart or to the identified peptide?
So it will be related exactly to this identified peptide.
Ah, okay. So for instance, as you in your table, you have 2 times oxidized peptide,
yeah, exactly.
then it would be 16 Dalton to the oxidized peptide, for instance.
Yeah.
Okay. And with labeling is the same?
Yeah.
Okay, thank you.
Okay. So I think before I will just 1 second say something about the topic that's very hard, that's the Perseus plugins. You all know that we are on plugin programming, and you should really check this website. And there's all the materials, and also if you want to fully participate, you should install some requirements. So, for example, the development environment. So you need your laptop, and I'm volunteering to stand in the corner here in the coffee break today and tomorrow. And just if you have any issues with this, then please come to me. Or if you don't have issues and you just want to chat. So.